This is the MyHeart.net podcast. This show is produced by Dr. Philip Johnson in conjunction with VitalEngine.com. Please welcome your host, Dr. Alain Bouchard of Cardiology Specialists of Birmingham, Alabama, at St. Vincent's Medical Center, part of Ascension.
Welcome to our podcast on the COVID vaccine. And with me, we have our special guest, Dr. Leland Allen, who is the Chief of Infectious Disease at St. Vincent in Birmingham, Alabama. Leland, welcome to our podcast, and thank you for agreeing to do this.
Thank you. It's my pleasure.
Yeah. Dr. Allen obviously is in infectious disease. He's been really in the forefront of this fight against COVID. I'm a cardiologist. I feel like more in the back seat and in the background primarily. So we really owe it, to you and the people that you work with to really help, our patients. So thank you for that.
Well, thank you.
So we'll talk about the COVID vaccine today, and we know that to date, there's over 167 vaccines that are being worked on, 8 of which actually are undergoing and are going or starting a phase 3 clinical trials. It seems like the U.S. has set up this Operation Warp Speed, which is basically a collaboration of the Department of Health and Human Services, the FDA, and other federal agencies to deliver 300 million doses of a safe and effective vaccine by 2021. Also, pharmaceutical companies are involved with this, and particularly AstraZeneca, Johnson & Johnson, Moderna, Novavax, Pfizer, just to name a few. FDA says that for any vaccine to be approved, it will need to prevent infection or decrease the severity of at least 50% of the people vaccinated. The goal is to inoculate enough people with a vaccine that immunity spreads throughout a community, the so-called herd immunity, so that 60 to 70% of the population would need to develop antibodies. So, Dr. Allen, if you could maybe kind of describe, for us, what is immunity and how does a vaccine work?
Well, the process of vaccination started back in the 1800s when Jenner first realized that people who had been infected with cowpox had no smallpox. And so he generalized this to believe that there was some sort of immunity that was given. And so the first vaccines were really fortuitously discovered. But the idea is that by giving some priming of the immune system, you prevent the development of an infection or development of complications. And that priming has taken various forms through the years. At first, it was giving a different infectious agent that provided some cross-reactivity or cross-immunity, and then progressed up through the polio vaccines, where they were using attenuated viruses to infect people, actually cause an infection, but with a virus that didn't cause as severe a disease. And then we started giving inactivated viruses and then various proteins to try to stimulate the immune system to provide some protection. And so the idea is that by preparing the immune system to face a particular pathogen, we prevent that pathogen from causing problems. So
that's obviously you're able to provide to provide some immunity to the individual. You bring in an antigen, and the body builds an immune response, builds some antibodies to it, and you have therefore immunity against that antigen.
That's correct, and the immunity happens in different ways. I like to tell my patients when we're talking about the immune system that the immune system is not a monolithic, homogeneous system. It's a number of parts that work together, and each part has a particular role. And so I think of the immune system kind of like I think of the Army, the military. There are different roles that different sections of the military play. And so you have an Air Force, you have an artillery, you have ground troops, and each of those provide some different function in the overall functioning of the military. For the immune system, there are lots of different parts of the immune system, and different parts of the immune system are tasked with protecting us from different types of pathogens. And so people think about antibodies with the immune system, and that's one aspect of the immune system. Sometimes developing antibodies alone doesn't provide any effective immunity. There are other parts of the immune system, the cell-mediated immunity or the cellular immunity that involves T cells and natural killer cells and so forth, also provides some very important protections against certain pathogens. And so one of the goals of vaccination and immunization is to try to target the parts of the immune system that are specifically responsible for defending us against certain pathogens. And so, for instance, with viral infections like the SARS-CoV-2 virus, that antibody production will be important, but the development of cell-mediated immunity is going to be very important also, both for managing acute infections and also for providing long-term benefits. And that's been one of the challenges in vaccines through the years, is that we can generally make people develop antibodies against things. For instance, in HIV research, we were able very early to create antibodies in people against various parts of the HIV virus, but they weren't protective against infection or protective against the development of disease. It requires a more comprehensive development of immunity. And so far, we haven't been able to do that. And so one of the challenges in developing any vaccine, and especially a vaccine for COVID-19, is truly defining what it takes to be immune to COVID-19 and how we can stimulate those parts of the immune system. And some of that is work that's ongoing. We know in general that coronaviruses overall require antibodies and cell-mediated immunity, but we don't know how much of each is required to give good immunity, and we certainly don't yet know how durable that immunity is going to be. So it's a challenging field to develop a vaccine for this, and we're doing a lot of learning on the fly.
So we're trying to develop some immunity or mechanism of defense for the individual. What do we call, what is herd immunity?
Well, herd immunity is actually the concept that if enough people in a particular community are immune to a certain disease, that there just won't be any passage of that there's not going to be enough susceptible individuals in that community to sustain transmission of a virus. And so even the unvaccinated people in a community that has good herd immunity probably are going to be protected just because the virus isn't going to be present. This was one of the issues, for instance, with measles and measles vaccination in the past, that we had developed pretty good herd immunity in the population, but vaccination rates have waned over the past decade or so because of people wanting to opt out of vaccination, and also influx of unvaccinated people. And so last year we saw some pretty good outbreaks of measles where we had not seen that before, because all of a sudden there were more susceptible people, and the virus was able to get into the non-susceptible population, and there were enough susceptible people to sustain transmission. So herd immunity is very important in the overall protection of a population, meaning that we don't have to vaccinate 100% of the population as long as we can get a good portion of that population, will be very effective.
There's obviously some individuals, some patients that have that are immunocompromised, or even the newborn that are less than 6 months old that are not really candidate for vaccination. So it's very important to have enough of the population vaccinated so that they provide this kind of immunity, and the disease will not propagate to the patients that are most at risk and cannot.
That's absolutely correct.
Yeah.
So taking a vaccination, just in the broader sense, taking a vaccination is not just an individual choice. It is a choice that you are helping to provide some greater good to the community.
Yeah, and particularly important for us with our cardiac patients. Also, a lot of them are immunocompromised, just because they are so sick. Well, we talk about COVID. I mean, right now we have probably just in the United States 5.5 million patients that have been infected with COVID, and obviously we have a very large population. Certainly, we could get some immunity if we have enough people infected, but we sure wouldn't want to have millions of people, more people being infected. Number one, I think it would really kind of increase the number of patients would be at risk of dying, would totally overwhelm our system, and therefore the very important, vaccination program that is about to get started, and talking about the COVID-19 vaccine. 8 of which are currently in clinical trial. Could you tell us, Leland, about the vaccine? I mean, there's several ways, multiple ways to make vaccines, and obviously you've described a little bit earlier using whole virus that have been weakened or inactivated. But there's many other new ways now to make vaccine, some of which actually are going to have the benefit of it. I mean, if you think about it, the first infection with COVID-19 was in China in December 2019. Already in January, they had deciphered the genome of the virus, and vaccines were starting to be made in March. So what are the different kind of vaccines do we have for COVID-19?
There are several different ways, and some of them are quite fascinating from a technical standpoint. Obviously, sort of the oldest, easiest way, and there are a couple of these trials going on in China where they are simply taking the virus and killing it or inactivating it and giving that to patients as a vaccination. So just the whole killed virus, and that's been done with other viruses with pretty good success in the past. But some of the newer technology involves genetic manipulation. And so, for instance, there are a couple of studies out there looking at taking genes, specific genes from COVID-19, and putting them into other viruses that aren't as bad. And specifically the gene or the protein that everybody is using is this protein called the spike protein. And if you've seen a cartoon or if you've seen an electron micrograph of a coronavirus, that it has this halo around it, the crown, which is where we get coronavirus. And that crown is made up of a specific protein on the surface called the spike protein. And it induces a pretty good immune response, and it's one of the things that's required both for attachment of the virus to susceptible host cells, but it's also a good target for antibody immunity. So what they have done is they have taken the gene that encodes the spike protein and transfected it into adenoviruses, and adenoviruses are common human viruses that do cause some disease, but typically more in the cough and cold kind of problem, and not in the immunocompetent population. Adenoviruses are not generally considered to be a huge problem as far as mortality. And so they take an adenovirus and have it specifically express this gene for the spike protein and give it to patients. And therefore, the virus infects the patient and causes the creation of this spike protein, which the body then recognizes and develops an immune response to. And that's there are a couple of adenovirus-based vaccines floating around out there. And then probably the newest, neatest technology is mRNA vaccines. And here at St. Vincent's, we're going to be starting an actual trial, and this is the Moderna product, which is a vaccine where messenger RNA is actually injected into the patient. And messenger RNA is a secondary mediator in the protein synthesis pathway for basically any type of cell, whether it's a virus or a human cell. And it is how the genetic material sets out a blueprint or a template for actual protein to be produced. And so the messenger RNA in and of itself is not infectious, and it carries no risk of inserting itself into human genes or causing any kind of problems with manipulation of the patient's genetics. But instead, you just give this mRNA, it gets into cells, and it's the mRNA that also encodes the spike protein from the coronavirus. And so it basically causes our own cells to create the spike protein, which again is recognized as a foreign protein, and immunity is developed. And then the mRNA is degraded and it goes away, and it's not a problem. And so you get rid of the genetic material. You still have some immunity that hopefully has developed against the specific protein that we're looking for, and you don't run into the problems with genetically altered viruses that an adenovirus might actually cause a disease in the patient that you're giving it to. So it's really new and exciting technology. We don't have a human mRNA vaccine yet that has been approved, but we think that the technical challenges that have precluded use of mRNA vaccines have hopefully been taken care of, and we'll be able to do this. And this has wide-reaching implications for vaccine development going forward, not just for COVID-19, but for whatever else comes down the pike in the future.
So this vaccine, for example, the mRNA vaccine, was tested in animals in the preclinical studies, and then in phase 1 and phase 2, which I think were just published recently, right?
That is correct.
And basically it was able to produce a lot of antibodies that were detected, but we don't know how long these antibodies last, right?
Right. And so the big questions that will have to be answered in the trials, going forward over time, and these are the kinds of things, obviously, that only time is going to tell, is one, when we have antibodies and some degree of cellular immunity, is that going to protect the patient from the development of disease or protect the patient from infection? Because one of the things that happens with some vaccines is that a patient can still become infected with a particular pathogen, but the disease that it causes is much less severe, or the death rate goes down, even though there are infections. And so those are good. Obviously we'd like to prevent as many infections as possible, but if we're not preventing infections, but we're preventing death, that's a pretty good outcome also. So obviously we need to know if giving this vaccine and if creating these antibodies is going to prevent infection or reduce morbidity or mortality. And also we got to find out how long these vaccines are good for. We know that with a lot of vaccines, some vaccines, you get a vaccination and it's good for life. For instance, if you get two doses of hepatitis A vaccine, you're protected for life. But with other vaccines, this is why we have to give tetanus boosters, and this is why we have to revaccinate people for a lot of other pathogens, because the immunity wanes over time. So, those are questions that still have to be answered, and will only be answered over the next few years, really.
So in the phase 1 and phase 2, trial, they were able to detect a good response. Was there any side effects that were actually found? I agree, it's kind of a small number of patients, but still, that's the only experience we have.
Right. We think, at least with the Moderna product, that some GI illness and obviously some injection site problems were problematic, but we think it's going to be a fairly safe and well-tolerated vaccine. Obviously, again, it's been given to a very limited number of patients, and the Moderna trial, for instance, is going to try to enroll 30,000 patients. So we'll have a much larger sample size that we'll be able to detect less frequent side effects.
So the phase 3 clinical trial is starting, this week at St. Vincent. So maybe we should kind of discuss who is a candidate for this, and where do we go to get the information to enroll if we have some volunteers that are interested.
Sure. The candidates, obviously, if you're going to test a vaccine, the ideal candidates would be people who are at risk, because we're not infecting anybody. So we would like to see people who are in higher risk categories, both in higher risk for becoming infected or being exposed to COVID, but also people maybe who are at higher risk for developing complications. So we'll entertain anybody, but obviously we're looking to enroll people who are older, maybe who have some comorbidities that would put them at increased risk, like chronic lung disease or heart disease or some sort of immune deficiencies. And then obviously people who are in positions where they are more exposed. So obviously somebody who is sheltering in place in a cabin in the woods and hasn't seen another live body for 3 months is probably not the ideal candidate for us. So
healthcare workers would be
healthcare workers would be,
because they're exposed.
Healthcare workers would be great. Teachers, unfortunately, are going to be in that higher risk category. People who are considered essential workers, but also people who are choosing not to shelter in place. We all have friends who are hand waving the recommendations to social distance, and they're going forward with their life full speed ahead. And not surprisingly, those are the people that we ultimately are seeing in our clinics and in the hospital. So those are people that would be better suited for the trial. Again, we have a screening process. If you're interested in the trial, you can send an email. Right now, you can send an email to stvhsclinicaltrials@ascension.org, and somebody will contact you. Once we actually get the study up and running, there'll be a website that you can get to through Moderna. But we closed down we had some logistical problems getting launched. We thought we were going to get this going a few weeks ago at St. Vincent's. And so we had a website up and going, but we took it down because we felt it was the wrong thing to do, to have patients expecting to be vaccinated and then keep pushing back. But we actually got our product day before yesterday, so we're ready to go, and we'll start seeing patients this week.
Well, we're talking about vaccination, how important it is, and particularly for our cardiac patients. Obviously, you've been treating these patients every day, Leland. Can you tell us just kind of briefly what kind of cardiac complications that you've seen actually in the patients you treated, and other complications such as, pulmonary and so forth, and even with people that recover, what kind of sequelae do we find months later?
Well, one of the interesting things about this is that it is most certainly not just a respiratory disease. This is a whole patient disease, and you can find complications in every system in the body. And some of those are complications of just the general inflammatory state. We know that anytime somebody gets an infection and the body's total inflammatory system is revved up, that increases complications in all other systems of the body. For instance, with cardiovascular disease, the inflammatory state in the body is a prothrombotic state, and so patients are more likely to develop thrombosis, so an acute coronary syndrome. And that is not just for COVID-19, that's for just about any viral disease. So this is why we see problems with during flu season. You see cardiovascular complications go up in flu season. But we've seen a fair number of people that have pulmonary embolisms, deep venous thrombosis, again related to the prothrombotic state. The latest thing that came out last week or 2 weeks ago is there's a Boston Red Sox pitcher that developed myocarditis from COVID-19, and there are several case reports of patients developing an acute myocarditis, cardiomyopathy involving COVID-19. There was a study in, I think, JAMA Internal Medicine that came out last week or 2 weeks ago that looked at ST elevation MI mortality during the COVID epidemic up in New York, and what they found was that over the past month or so, that the numbers were staying the same, but the mortality in ST elevation MI had gone up. And one thing they didn't do in there was really stratify for who actually had COVID-19 and who didn't. But taking the population, it was alarming to see that it seemed to have a higher mortality. So I think from a cardiovascular standpoint, in painting with the broadest brush, it's never good if you have underlying heart disease to get sick with anything, because underlying congestive heart failure, angina, all of those things are going to get worse because of the metabolic strain that having a systemic infection puts on the body. And when you couple that with the significant hypoxemia that these patients sometimes develop, that just is a recipe for disaster from a cardiac standpoint.
Have there been any autopsy studies showing the actual virus in the cells of the myocardium or the vasculature?
I don't know about the myocardium. I know that there have been some suggestions that there's an actual vasculitis, and that's been one of the suggestions about the pathogenesis of the pulmonary disease, that it's actually a pulmonary vasculitis that happens. I'm not sure about myocardium, though. I have not seen that. It certainly doesn't mean it hasn't happened. It's one of the amazing things over the past 5 months is the amount of just the amazing output of literature. Some of it just fascinating and amazing, and some of it absolute junk that has been published regarding this. And I don't know anybody who can keep up with all of it.
Yeah, probably impossible. I know we're wanting to talk about prevention primarily, but we want to focus also and emphasize how important this is. A deadly infection, and it's so important to prevent it. There are some, treatment that have been, coming to the forefront. Unfortunately, the studies have been also, difficult, but it looks like remdesivir, if given early, can shorten the disease. It looks like steroids has a role. How are we doing with the treatment of COVID-19?
Well, obviously there was a lot of enthusiasm early on for Plaquenil, for hydroxychloroquine. That has not been borne out in prospective trials. And there have been some retrospective studies that have been done. Obviously, retrospective studies are never as good as a prospective study because it's just impossible to control for everything. And in this case, the studies that were done just didn't control for some things. For instance, there was a big study of Plaquenil that came out of Henry Ford Hospital in Detroit that retrospectively did show a benefit to taking Plaquenil. Unfortunately, that study didn't control for steroid use, and steroids, I think, have been shown pretty conclusively to be helpful. So the prospective studies done with hydroxychloroquine have not shown any benefit. There's a concern, obviously, again, from the cardiovascular standpoint of hydroxychloroquine and prolonged QT syndromes, but that surprisingly has been not a huge problem either, not as bad of a problem as it probably could have been. There were some people early on, myself included, who used hydroxychloroquine in combination with azithromycin. That is really throwing water on a drowning man in terms of arrhythmia potential. And so I think the FDA put out a pretty sharp warning about don't do that because that's two drugs that can prolong the QT. So I think the Plaquenil ship has sailed. I don't think there is good science that would reasonably support its use. Remdesivir had some promise in the beginning, but again, I think the more we use it, we're not seeing just miraculous turnarounds. And some of that is figuring out the timing on when to give remdesivir. Obviously, with most antivirals, the earlier you give them, the better the response is going to be. And so ideally, the time to give remdesivir is probably before the patient is symptomatic. It just theoretically, from looking at how antivirals function, obviously that's not going to happen. So we have been trying to limit our remdesivir use to people fairly early on in the disease process, 7 to 10 days after the onset of illness. And beyond that, I don't think it's really going to help. And we haven't yet seen a mortality benefit that has been significant with remdesivir. So I think the role of remdesivir is still being determined. There were a couple of other drugs that were given. One of the things that we've noticed about the clinical course of the disease is there's an initial phase where this is probably a viral syndrome, like any other viral syndrome that we've all expected. The patients feel terrible and all this, and this is caused by direct viral circulation. But there seems to be a second wave of the illness that comes on 10 to 14 days after the initial wave, and that is probably less of a viral problem and more of an inflammatory problem, this hyperinflammatory syndrome. And so there was some thought early on that using inflammatory modulators, and the one that got a lot of use was tocilizumab, which is an inhibitor or blocks the IL-6 receptors. And it was thought that this would blunt this hyperinflammatory syndrome, and I've used a fair amount of it. My clinical experience on it was that it helped patients for about 12 hours, and then it went away. The company that makes it was actually doing some studies, and I think 3 weeks ago, they stopped all of their studies after an interim analysis because they found no benefit to it. So I think that's probably not going to end up being an option. There's some other monoclonal antibody-based products that also deal with the interleukin blockade that might theoretically have some benefit, but there are none of them that are in common use right now. I think, going back to what works, I think being able to deliver oxygen as best as we can is important. And so using high-flow oxygen in these patients and being able to maximize their oxygenation without putting them on a ventilator. And so in our institution, we're using BiPAP, but run through a ventilator circuit. So it's a closed circuit, and we're not aerosolizing virus into the environment. Has been very effective. And then prone ventilation has actually been very effective at improving oxygenation. Even in patients that aren't on a ventilator, just having them lie on their stomach changes the hemodynamics in the lungs, and it really does seem to improve oxygenation. And then, of course, steroids. We think there's a pretty good benefit to steroids.
So the treatment are not that great. And so if we could prevent, that's probably a whole lot better.
That is absolutely correct. This is a disease that you don't want to get.
Right. Now we have, obviously several patients have gotten the disease. It was very mild. They've recovered from it. Now we're not that far into this pandemic yet, but are there some cases where the patient got reinfected and got sick a second time, or we just don't have enough data?
Well, obviously, that's an important question to ask because it really circles back to the question of whether or not vaccination is potentially effective. Because if having the actual disease doesn't protect you from getting it again, then providing an effective vaccine is going to be extremely challenging. And it's actually been kind of a more difficult process than you might have expected to identify when patients get reinfected for a couple of reasons. One is the whole problem of how do you test for this virus? And I know testing is a buzzword that has just saturated the news cycle from almost the beginning, but testing has really been problematic because the testing is challenging from a logistical standpoint. It is challenging from an availability standpoint, and it's challenging from a sensitivity standpoint. The tests that we are using now, we're finding may be more sensitive than we need. The PCR-based tests can remain positive for long after the patient, maybe a couple of months after the patient has completely recovered from their illness. And we don't exactly know how to interpret a positive PCR-based test. The question is this patient sick with this virus? If they come in and get retested, say my employer won't allow me back to work until I have a negative test. Well, we do this test. Patient's been completely asymptomatic for a couple of weeks, but their test is positive. Does that mean that the patient is infected again? Probably not. Does that mean that the patient is contagious to everybody else, which is the real reason that we would want to know, before letting somebody go back to work, do they pose risk to their environment? And the answer to that is we don't think so, but all we have is this positive test. The test that we used to diagnose them to begin with is still positive. And so what we think is that what's being picked up on that test is not replication competent virus. It may be viral residue, or it may be attenuated virus, but using some other mechanisms, and the CDC has referenced this in a big update they did about 3 weeks ago to their return to work criteria, that they have some data that the virus is probably not present in a contagious form 7 to 10 days after the onset of symptoms, even if the test remains positive. So we haven't seen, I haven't personally seen people who I think, have gotten over the disease and gotten sick again. I've seen plenty of people that we treated early on in their viremic phase. They got better, see them 2 or 3 days after onset of symptoms, they get better, and then they get readmitted a week or 10 days later, now with this hyperinflammatory stage. And so we see that, but we haven't seen anybody that was infected back in April that's come back in July with, I was fine and now I'm sick again. What we do see, though, are patients that get sick and really have a prolonged convalescent period, that they get better in terms of their fever's gone away, maybe their cough is better, maybe their oxygen requirements are less, or maybe they're not requiring supplemental oxygen, but at the same time, they have a prolonged fatigue, their exercise tolerance is gone, they might have a persistent cough that lasts for months. It's just kind of a mild, nagging, hacking cough. And, these people may also have some prolonged increased risk of thromboembolic events. Again, that's the kind of thing that we'll find out over the course of the next 3 or 4 years, where we have long data sets to look at. But we think that patients really can have a prolonged convalescent phase that takes a long time to get over, and it's not that they're persistently viremic, it's just that the virus and the inflammatory response to the virus has done enough damage to their body that it really takes a toll.
Yeah, there was this study, I think it was published in JAMA, that you probably saw it, in Germany, where they performed cardiac MRI, finding some evidence of scarring in some of the patients
That's
that
right.
had recovered, from COVID.
That's right. And that probably represents I would say, a subclinical myocarditis that we just didn't recognize because of all of the other stuff that was going on in the patient. As you are well aware, not everybody gets a cardiac MRI. That's a challenging thing to do, especially with patients on 50% closed face mask oxygen. So, these are incidental findings that we see in patients that then trigger us to say, okay, what was going on back when the patient was really sick? And circle back around and say, okay, well, maybe this is a problem that we just didn't recognize at the time.
Right. Coming back, I'm sorry.
I was going to say, this is just all the things that require time. We would like to have all the answers up front, but really you have to have the lens of time in order to really determine what was going on and what's really going on with the patient.
A year or two, probably.
Yes.
Coming back to the test, it seems like it would be more helpful to have a less sensitive test to be able to maybe help more people. There's this mention about this saliva test that could be implemented much easier, a little bit less sensitive, but maybe it could be more helpful in this situation.
That is absolutely correct, because you really want a test to answer two questions. One is, does the patient have the disease? And two, is the patient contagious? With coronavirus, those are the two most important questions to answer with the test. And so in our case, our current testing doesn't always help us answer that second question. Well, it doesn't answer the second question at all. And the first question is a challenge just for logistical reasons. Here at St. Vincent's, we're very lucky in that we have enough capacity that we can get pretty rapid turnaround on tests. But if you go to a walk-in clinic and get a coronavirus test and they're not able to provide you with an answer for 6 or 7 days because of the turnaround time on their testing, why did you even get the test? That's not helpful for anybody. And so the salivary test that you talked about, the advantages to that test are that it's cheap, and that I think at least one of them that I've heard of has about a 15-minute turnaround time with minimal investment in labs. So you could at least get an answer back very quickly. And it does have a lower sensitivity, but as I mentioned earlier, that might not be the worst thing in the world. You have if it's only detecting high levels of viral burden, those are the patients that are most likely to be contagious. And so those are the patients that you would really want to concentrate on keeping away from susceptible individuals and putting them in quarantine as much as possible. And obviously that's all a theoretical discussion because none of that has really been borne out in clinical trials. But if you had a test that was cheap and quick and was able, especially to identify people that are at risk of transmitting to others, that would be the ideal thing to do in big populations of people like school-age kids or nursing home patients where you've got a high potential for spread, and so you can isolate those people from the group quickly.
Well, Leland, I treat a patient, a population at risk, and we have this COVID, and I'm so happy that the trial is starting, with the vaccine. But obviously, we're not going to find, any, answer for the next several months. I mean, possibly have some answer by the end of the year. But in the meantime, there's the flu season, coming on, which is already, if we look at 2018, 2019. The flu season was considered moderate, and yet it affected 35 million Americans who fell sick, with almost 500,000 were hospitalized, and over 34,000 people died. And this occurred while 170 million doses of vaccine were administered. So how do you think this COVID flu season is going to play out?
Well, even taking COVID out, asking anybody in August how the flu season is going to play out is just about like going to Las Vegas with a pocket full of money. Just don't know. We always hope that it's going to be a mild flu season, but you never know when this is going to be the bad one. And so I would say that we could have the best case scenario would be this is a mild flu season. And the social distancing measures that everybody's putting into place for COVID are also going to secondarily impact risk for transmission of influenza. And at the same time, we hopefully continue to see a downward trend in COVID infections. And so the best case scenario is this ends up being a pretty mild season for cold and flu. The worst case scenario is that we have a bad flu season, a bad flu strain come through. We have bad mismatch between the vaccine strains and what actually ends up circulating. And the coronavirus picks up also at the same time. And then we have a situation where we've got probably not just an additive effect, but probably a multiplicative effect of the two, because what we saw very early on in the coronavirus epidemic was that people were worried about COVID, and so they were delaying care for other problems. You all saw this in cardiovascular disease,
yes.
And it was seen across the board that people just couldn't or didn't go to the doctor for problems that should have been seen, and it became problematic. And so, if we're having a bad coronavirus outbreak, then people are going to be less willing to go seek health care. Healthcare may not be available. If we have capacity problems in hospitals, then it really could be an exponential problem, and we could have a really bad cold and flu season. So I don't know. I'm praying for the former.
Yeah, well, I'm happy to see that at least there's a lot of things in place to prevent infection. For example, the mask, the distancing, the washing the hands. I mean, there's been such a strong emphasis to prevent the COVID virus, but it's also a very great mechanism to prevent acquiring the flu.
Absolutely, they're different, totally different viruses, but the transmission properties are the same. It's respiratory droplets, it's contact with infected surfaces. And so the things that we do to protect ourselves from COVID are the things that we need to do to protect ourselves from influenza. Plus, we have the added benefit of being able to vaccinate against influenza. And it's not, it's it's a terrible vaccine in the grand scheme of things in terms of its protection, but still it's effective at reducing morbidity and mortality. And I just can't strongly urge people enough to get a flu vaccine.
Yeah. So let's talk about the flu vaccine a little bit. I mean, there's also different forms of flu vaccine. You have like the egg base. I don't know if it's still used anymore. There's the cell base, and the one that we're talking about with COVID with recombinant DNA. So what, in the flu shot that we give, what's the most popular and what works the best?
What works the best is the one that actually gets out of the vial and into the patient. As one author I read said that a vaccine is worthless until it becomes a vaccination, and that is the truest thing I've ever said, because we just, the challenge of getting something into people is really helpful. And honestly, I think they're all pretty well effective. Equally, one has not been shown to be more effective than the other. And I think if you go to your doctor or you go to your local pharmacy, you're going to get what they have rather than that. Now, the vaccine that we generally are using now is a quadrivalent vaccine, which means it protects against 4 different types of flu. And over the last few years, we've started to present a higher dose vaccine for patients that are at high risk of flu complications, patients over 65, patients with other comorbidities. And that's probably going to turn out to be a better vaccine in that higher risk population. But again, the important thing is to get a vaccine.
Right. So if you're 6-month-old or older, you should get a vaccine, but can you get all the vaccine? I mean, some of the vaccines, you have to be 18 years old.
The vaccines are available for anybody now. Live virus vaccines, which there used to be a live flu vaccine called FluMist that they have now taken off the market. So that was a live attenuated virus, and we didn't recommend it for people who had any kind of immune compromise. But as far as the vaccines that are available now, you should talk to your doctor, but basically everybody is eligible for it.
Yeah, well, a lot of family physician, practice telemedicine. Certainly during the flu season, it's time to reopen the door and be able to give the flu shot. Or are we going to have we're going to have drive-through flu shot, or how do you see this kind of work out during this season?
Interestingly, flu vaccine is available right now, which is earlier than it's ever been. But part of that is because early on in the COVID epidemic, even I think back as early as January and February, people realized that this was going to be a problem for this upcoming cold and flu season. And really, just as an aside, manufacturing flu vaccine really is a year-long process. It is something that there are already people now starting to work on the 2021-2022 vaccine. So just trying to identify what strains we're going to put in the vaccine and so forth. But they identified very early on, and so there was a push by the government to really ramp up production so that we got the vaccine out, didn't try to avoid any hiccups, and produce more vaccine this year than we had before. So as a result, we have vaccine available right now if you want it. I think you can go to your local CVS or Walgreens and get it now. It may be a little early because flu vaccine is like some other vaccines in that the immunity wanes. And so it may be better to wait until next month before you get it, because we really haven't seen much flu circulating. But I think there is going to be plenty of vaccine available in multiple venues. And as far as a drive-through vaccine clinic, I haven't heard of one, but I suspect that somebody will come up with that product pretty quickly, especially if there's any money to be made from it.
Well, certainly it will be very important for us healthcare workers,
right,
as well as our patient, to get the flu shot.
Right.
I mean, I can't imagine, getting the flu. How, as a physician, how are we going to be able to tell whether someone has got COVID or someone has got the flu? I mean, it's really going to be difficult.
It will be difficult. Obviously, with any viral illness, whether you're talking about COVID or flu, there is a lot of art to that medicine in addition to the science. And so you sort of combine the art of bedside diagnosis and history and physical with looking at the epidemiology, literally what is circulating in the community right now. And it's not completely ruling out one or the other, but it's going to be looking at what's circulating in the community. In addition, we do have pretty good flu testing right now, and a lot of the platforms that allow COVID testing right now are going to have a product that will come out in the next month or so that will have a combined COVID and flu test, so you can do one swab and get an answer for both. I know that Cepheid is going to have that product, and as a matter of fact, they're going to quit making standalone COVID tests available and only make a hybrid COVID influenza test to try to sort this out, to help answer that question appropriately. But again, the issue is going to be testing availability and turnaround time on it, and that's been a bugaboo in a number of cases.
Right. So for the patients that are going to sign up for the COVID testing, it's not going to be a problem to have the flu shot?
Oh, no. We are absolutely encouraging patients to have flu shot and any other appropriate vaccinations.
Maybe we'll be able to give COVID vaccine by the end of the year, you think, or?
Well, the FDA has set a goal of having a vaccine in place by the end of the year. I think that is very ambitious, but there has been a tremendous amount of money and labor that has been poured into this. So we may have a vaccine, and again, only time's going to tell how effective and good it is. And I suspect what we'll end up doing is the FDA will end up approving multiple vaccines. As you said at the beginning, there are multiple vaccines that are in trials right now using different methodologies. And I think that they're going to approve as many as they can so that we get some real world experience with this. And then over the next 5 to 10 years, something will shake out. This one works. This one really doesn't. This one's better than that one. And we'll just see.
Well, it should be interesting to watch also what happens in Russia, where they have approved the vaccine without any phase 3 clinical trials. So,
Putin's vaccine. Yep.
Right, we'll find out. So, a lot to find out. We don't know whether we're going to need a booster or not. We don't know whether we're going to need a COVID vaccine every year, the way we need for the flu. Of course, we need it every year with the flu because it changes all the time.
It does change.
I don't think we should expect this COVID-19 vaccine, SARS-CoV-2, to change that much.
The interesting thing about any vaccination, and I made this point up front when we first started talking about trying to come up with a vaccine, is that coronaviruses before COVID and before SARS before it, coronaviruses were well known as a common cause of the common cold, and that's where we knew coronaviruses from. I mean, coronaviruses, rhinoviruses, some adenoviruses are really common cold viruses, and so what we are basically to put the vaccine challenge into perspective, what we are basically trying to do is since the beginning of the year, come up with a vaccine for the common cold. And so in medicine, we know what the challenges are in doing that. But we've really made some progress, which is just astonishing considering how challenging dealing with the common cold has been forever.
Yeah. Well, Leland, obviously it's going to be very interesting to start the clinical trial on the COVID vaccine. It's going to be an interesting season, and
let's hope not.
Yeah, well,
interesting is never good.
Interesting, maybe for the better, but I don't see a whole lot of sleep in your future. You're going to be a busy man within the next several months, and we appreciate so much what you do, the guidance, and the taking care of all of our patients. So thank you very much for this,
thank you,
Dr. Allen.
It's been a pleasure. I appreciate it.
Thank you so much.
All right. Goodbye.
Bye-bye.
To learn more from our team of cardiologists, please visit us at MyHeart.net. You can also follow us on social media by searching MyHeart.net on Facebook and Twitter. And be sure to subscribe to this podcast so you don't miss our next episode.